Welcome everyone, and thank you for those joining us live wherever you are in the world, and to those listening in afterwards to this FIP event. I'd also like to warmly welcome our guest speakers today, who I will shortly introduce. My name is Chris John. I am the FIP lead for the COVID-19 Digital Event Programme 2022. I'm also FIP lead for Data and Intelligence. FIP's vision is a world where everyone benefits from access to safe, effective, quality and affordable medicines, as well as health technologies. Our mission is to support global health by enabling the advancement of pharmaceutical practice, sciences and education. We are pleased to be delivering this event, which is episode number 3 of a series of 6 events of our COVID-19 programme. And the programme's title is How Has COVID-19 Changed Us? Pharmacy and Pharmaceutical Science Perspectives. Today's event is about therapeutic advances against COVID-19. Pharmacists and pharmaceutical scientists have an important role to play in the continued response to COVID-19, as well as preparing for future pandemics. This series of events will be delivered from has been going since April, but will be delivered up until December this year. Just going to move on to some announcements. We always do this at every webinar. It's being recorded, so if you want to revisit it, it will be on our website today, and it's also being live streamed via YouTube. The website address, in case you don't know it, is there on the screen now. And during the event, you may ask questions please do use the Q&A box. What we will do is answer the questions in a panel discussion at the end. Excuse me. Please also do use the chat box, just to say hello and network with your colleagues. You're welcome to provide feedback on any of FIP's webinars at webinars@fip.org. And please do become a member of FIP, and you can find out about membership in the email address just at the bottom of the screen there. So on to today's programme, Therapeutic Advances Against COVID-19. I'm your moderator, so I will be keeping the slides going, keeping the event going. We will start off with Dr. Syed, Shahzad, Hasan, who will talk about currently available treatment options for COVID-19. And then we will move to Professor Trevor Jones, who will talk about future treatment options for COVID-19, and finish off with a short panel discussion. So please do use the Q&A box and close off in around an hour. Today's learning objectives are to explain why medicines are available that target the virus or provide symptom control in COVID-19, discuss which medicines will be effective against new variants, describe what other medicines are available and what is the evidence for them, and to list medicines in the pipeline for treating COVID-19. So on to our first speaker. I'm delighted to welcome Dr. Shahzad Hasan, who's the Senior Research Fellow at the University of Huddersfield in the UK. He's also Senior Lecturer at the University of Newcastle, Australia. He's got over 15 years of experience as a pharmacist and on academic staff, and a researcher in Pakistan, Malaysia, Australia and the UK, as well as being a section editor of the Journal of Pharmaceutical Policy and Practice. So without further ado, I'll hand over to Dr. Hasan. Thank you very much.
Thank you very much, Chris, for the introduction, and FIP for giving me this opportunity to speak at this FIP digital event. Today in my presentation, I will be giving an overview of drugs that are currently available for the treatment of COVID-19, focusing on efficacy and safety of these drugs. We know that there are several drugs that have been recommended as potential treatment options or suggested as potential investigational therapies for the treatment of COVID-19. These drugs can be categorized into drugs for the treatment of COVID-19 infection, which can be further classified into repurposed drugs, drugs that have been used for other medical conditions and was repurposed for the treatment of COVID-19. And then we have some novel drugs as well. Then we have drugs for a critical or severe form of COVID-19, for example, dexamethasone or tocilizumab. Then there are drugs recently used for the prophylaxis of COVID-19. And then last category is vaccines for the prevention. I will be focusing on drugs for the treatment of COVID-19 used in the clinical practice or have been approved by the regulatory bodies in the UK or elsewhere. In the next slide, I will be going through the individual drugs. We know that in March 2020, the recovery trial was established as a randomized clinical trial to test a range of potential treatment for COVID-19. If you look at the treatment options available now for the COVID-19, corticosteroids, for example, dexamethasone, were found to be effective, the first effective treatment actually for a moderate or severe form of COVID-19. In recovery trial, a total of more than 2,000 patients were randomized to receive dexamethasone, 6 milligram once a day for by mouth or by IV injection for 10 days. And this drug was compared with the usual care alone. Dexamethasone reduced the death by 1/3 in ventilated patients and 1/5 in other patients receiving oxygen only. There was no benefit of giving dexamethasone to patients who did not require respiratory support. Based on these results, one death would be prevented by treating around 8 ventilated patients or around 25 patients requiring oxygen alone. We know that there is various corticosteroids preparation available in the market with different potency and duration of action. Unfortunately, results for other corticosteroids such as methylprednisones were not promising. There are several reasons for that. In the next slide, I will show you the meta-analysis that we have actually conducted, to find out the reasons why other corticosteroids, were not, effective in treating severe or moderate form of COVID-19. We actually, synthesized the evidence using the RCTs, to treat or to, COVID-19 or prevent, deaths or mortality in COVID patients. The pool mortality, all-cause mortality was not significant when we compare methylprednisolone with the control, indicating no mortality benefits. This could be due to the fact that most of the trials, there were 5 randomized control trials. Most of these trials actually used low-dose methylprednisolone without actually determining the equipotent doses. Therefore, to help clinicians, we propose dexamethasone equivalent dose estimation for methylpred therapy in COVID-19 patients, as you can see in the figure. In most of these trials, they actually use the same low dose methylprednisolone, the information they got from the recovery trial. So in determining the equipotent doses for an acute short course of corticosteroid, the biological half-life of steroids should also be accounted for besides the potency factor. A short duration, we found that in some of the trials, a short duration of a high dose methylprednisolone can be a promising alternative to the low dose dexamethasone therapy in severely ill patients with COVID-19. Another corticosteroid, in the next slide, the inhaled corticosteroid. After the success of this recovery trial with dexamethasone, inhaled corticosteroid budesonide was tested in mild cases of COVID-19 in principal trial, the same group who conducted the recovery trial, but in the community setting. They used 800 microgram twice daily budesonide for 14 days, and they measured the time to clinical recovery as an outcome. The trial reported that inhaled budesonide improves times to recovery with a chance of also reducing hospital admissions or death, but that findings were not statistically significant in people with COVID-19 in the community setting who are at higher risk of complications. There was no evidence in STOIC trial, another trial, of a negative effect of budesonide on SARS-CoV-2 viral loads, because initially there was a fear that these corticosteroids or immunosuppressive therapy actually increases the risk of this viral loads. And in principal trial as well, there were no concerning safety signals for inhaled budesonide. Recovery trial concluded or recommended that inhaled corticosteroids can be considered for patients with COVID-19 who are at higher risk of complications in the community. We also, in the next slide, will show that we have also conducted, several meta-analysis and systematic reviews synthesizing the evidence around the use of corticosteroids. If you remember, in March 2020, the WHO recommended against the routine use of systemic corticosteroids in the clinical management of COVID-19, citing the fear for the delay in viral clearance and other potential harms. As a result, there were concerns over the continued use of inhaled and oral corticosteroids in patients with asthma and COPD. But with our experience in dealing with the other, escalating, safety concerns with, other drugs such as the RAS inhibitors or blockers, our research team was, the first few to actually synthesize the evidence on the use of these drugs in COVID-19 patients. Our risk benefit assessment led us to conclude that the benefit of continuing corticosteroid treatment outweigh the certain risk in the context of the COVID-19 pandemic. We found a pool mortality estimate among COVID-19 patients with ARDS, the severe form of COVID-19, across all countries as 39%, but only 28% in patients with reported corticosteroid use, so almost a difference of 11%. Our systematic review with meta-analysis provided evidence of the effectiveness of low-dose corticosteroid therapy in reducing the risk of mortality in COVID-19 patients with severe course of illness, including those who develop ARDS. The WHO, if you remember later, overturned its previous recommendation to recommend systemic corticosteroid use in patients with severe COVID-19. Another drug, the tocilizumab, in the next slide, if you see, is an immunosuppressive drug used for the treatment of rheumatoid arthritis and other medical conditions. This drug is a humanized monoclonal antibody against the interleukin-6 receptor, is a cytokine that plays an important role in the immune response and is implicated in the pathogenesis of many diseases, including COVID-19. This drug was repurposed for COVID-19 and was effective in hospitalized patients aged 2 years and older who are receiving systemic corticosteroid and require supplemental oxygen, non-invasive or invasive mechanical ventilation. There are at least two big RCTs, the recovery trial and the remap cap trial, where the majority of their patients on corticosteroid showed lower 30-day mortality with tocilizumab use. A single dose with a second dose 12 to 24 hours later, if no clinical improvement was used in moderate to severe COVID-19 patients. So we see there is an improvement with the use of tocilizumab. There is a risk of secondary infection with the use of tocilizumab, but data from the RCTs, the big RCTs, showed a lower risk of infections and no higher risk of serious adverse events with the use of tocilizumab. Later, tocilizumab was granted an emergency use authorization to treat COVID-19 in the US in June 2021. The next antiviral drugs, the Remdesivir, is a broad spectrum antiviral drug, which was originally developed to treat hepatitis C and was subsequently investigated for Ebola virus disease before being studied as a post-infection treatment for COVID-19. The RCTs involving Remdesivir provided mixed results for the efficacy and safety involving COVID-19 patients. Wang et al. Showed no significant difference in negative viral conversion, clinical improvement, or clinical recovery, or even all-cause mortality at day 7 or even at day 28. But there was another trial which actually found a significant difference for all-cause mortality at day 14 and day 2. So there were mixed results, some positive and some negative results. In January 2022, recently, the Canadian component of WHO Solidarity Trial reported that in-hospital people with COVID-19 treated with Remdesivir had lower death rates by about 4%, and significantly reduced need for oxygen and mechanical ventilation compared to people receiving a standard of care treatment. In another trial, another big trial, Pine Tree trial, among non-hospitalized patients who were at higher risk for COVID-19, a 3-day prophylaxis course of Remdesivir, 200 milligram on day 1 and 100 milligram on days 2 and 3, had an acceptable safety profile and resulted in 87% lower risk of hospitalization or death than placebo. But again, the trial was sponsored by the manufacturer. In April 2020, the European Medicine Agency, EMA, provided recommendations on the compassionate use of Remdesivir for COVID-19 in the EU, later expanding the compassionate use of Remdesivir to those not on mechanical ventilation. The next antiviral drug, the Favipiravir. Favipiravir selectively inhibits RNA polymerase, which is necessary for viral replication. It is an antiviral medication used and approved to treat influenza in Japan. As an antiviral drug, Favipiravir has been authorized to treat COVID-19 in several countries, including Japan, Russia, Serbia, Turkey, India, and Thailand under emergency provisions. In the reported trials, the clinical recovery rate does not significantly differ between the Favipiravir group and the other antiviral drugs used for moderate or even severe patients. There is no significant difference in clinical improvement, even in mild to moderate patients with COVID-19. But some, again, there are some mixed results, some reporting positive results, some trials they are reporting negative results. Some trials they reported significantly higher improvement rate in chest imaging and faster viral clearance in Favipiravir arm plus interferon compared to other antiviral groups with interferon therapy. At the moment, results are inconclusive, and we are waiting for the results from principal trial, the same Oxford clinical trial unit, which is currently underway. The next antiviral drug, Molnupiravir, is an antiviral medication that inhibits the replication of certain RNA viruses. Molnupiravir was originally developed to treat influenza. Based on positive results in the placebo-controlled double-blind randomized controlled trials, Molnupiravir was approved for medical use in the United Kingdom in November 2021. In December 2021, the US FDA granted an emergency use authorization to Molnupiravir for use in certain population where other treatments are not feasible. The famous trial, the Move Out study, using the Molnupiravir, efficacy and safety of treatment with Molnupiravir, is started within 5 days after the onset of signs or symptoms in non-hospitalized, unvaccinated adults with mild to moderate COVID-19, with at least one risk factor for severe form of COVID-19 were compared with placebo in this trial, Move Out study. The risk of hospitalization for any cause or death through day 29 was lower with Molnupiravir compared with the placebo. In addition, the percentage of participants who were hospitalized or died through day 29 was lower in Molnupiravir group than in the placebo group. The results are promising, but these trials are limited in terms of numbers, so we are waiting for more trials, and it's been approved based on the Move Out study, the trials, the big trial in the UK and the US. The next one is the pexlovite, the brand introduced by the Pfizer, is a combination of two antiviral drugs. Remdesivir is an orally administered, antiviral drugs that actually inhibits the main proteases. It's co-administered with the ritonavir, which is required to increase the Remdesivir concentration to achieve the target therapeutic, concentration in the body. And it improves the pharmacokinetics of the Remdesivir. Evaluation of protease inhibition for COVID-19 in high-risk patients, the EPIC-HR trial, is a multinational randomized trial that compared the use of 300 milligram Nimeltravir plus 100 milligram of Ritonavir, which is used as a pharmacokinetic enhancer, and a placebo every 12 hours for 5 days in symptomatic, unvaccinated, non-hospitalized adults at high risk for progression to severe coronavirus disease. In the planned interim analysis of patients treated within 3 days after symptoms onset, the incidence of COVID-19 related hospitalization or death by day 28 was lower in pexlovite group than in the placebo group. The incidence of COVID-19 infection was on less than 1% in pexlovite group, and 0 deaths were reported, compared with 7% incidence in with the 7 deaths in the placebo group. So efficacy was maintained in the final analysis involving over 1,000 patients, but again, this trial was sponsored by the manufacturer. This drug was fully approved in the United Kingdom later that month, and in the European Union and Canada in January 2022. Last week, Pfizer reported that a large trial found, because this drug was again tested as a prophylaxis, and they conducted a large trial and found that it's COVID, the same Paxlovid was not effective in preventing, coronavirus infection in people living with someone infected with the virus. The trial enrolled about 3,000 adults who were household contacts exposed to an individual who, and had recently tested positive for COVID-19. So those who took the 5-day course were found to be 32% less likely to become infected than the placebo group. That rose to 37% with 10-day course of Paxlovid. However, the results were not statistically significant, and this possibly due to the chance. So at the end, so although the 2020, the pandemic, the COVID pandemic has given us unprecedented temptation to overrule the evidence-based approach, that we look at the big trials, the good quality meta-analysis, and the investigational therapies. We have, we should look at, the evidence and should not overrule the evidence-based approach in this process. As pharmacists, the guardians of safe medicine use, it's our duty to defend the evidence-based medicines even amid the pandemic and uncertainty surrounding the pandemic. So the last slide. So anyone working in a clinical setting, like pharmacists with patients, they need, this research skills to synthesize and process the evidence, looking at, the trial, the quality of the trial, the involvement of the patients, the details attached to associated with the trial, and at the end you have to decide whether the evidence is strong or is weak or in favor or against the use of any treatment. Beside, having this knowledge, theoretical and scientific knowledge. So we need this skill actually to know how to synthesize and process the evidence surrounding the drugs. So thank you very much. I will see what questions you will be asking.
Thanks so much, Dr. Hasan, for that comprehensive journey through the evidence. We've accumulated a lot of evidence in the last 2 years, and very important, as you say, that we try and adhere to it as much as we can. And I think we've come a long way since 2020. So thank you very much. Our next speaker, I'm delighted to say, is Professor Trevor Jones, who is a visiting professor at King's College London and a former head of research and development at Wellcome. He continues to have a distinguished career in the pharmaceutical and biotech industry, as well as in academia. He has held many FIP officer positions over a number of years, including being a member of the Board of Pharmaceutical Science, and he is a current member of the FIP COVID-19 Expert Advisory Group. So without further ado, I will hand you over to Professor Jones, who's going to take a little bit more of a future view of our therapeutic agents against COVID-19. So all yours, or the floor is yours, Professor Jones. Thank you very much.
Well, thank you, Chris. So if I could have the first slide, please. Going to talk, as you say, about future treatment options, because as we've just heard from Dr. Hasan, although we've got some choices, particularly in supportive therapy, like the anti-inflammatories and so on, obviously we don't have very powerful drugs that actually kill the virus, the antiviral drugs. And that's really where we have to focus more attention. But of course, we do have some very effective vaccines. But as you all know from your local use of these, their effectiveness doesn't last very long. And frankly, with the new variants coming along, particularly the new South African variants as well of Omicron, then we do need better vaccines than that. So if we look at the next slide, thank you. Right now, if we could take the vaccines we've got and put them into a more convenient form of administration, I know that would help immensely in many parts of the world, which some of you are from. We're actually trying to deal with the logistics of heat unstable vaccines is a real problem, the cold chain. Well, the Vaxart and the Oramed capsules are in the late stage of clinical studies. You'll imagine that with all the attempt to look at vaccine efficacy from the current vaccines, it's not so easy to do a trial on an oral vaccine when you know that you've got to try the existing injectable vaccines. But the Vaxart and the Oramed ones, I think within the next 12 months, we'll know whether it's likely that either of these or both of them could actually be a way forward. And then we've got a very different way of actually administering and probably in terms of price, affording the drugs. Meanwhile, a lot of people have been looking at intranasal delivery, and you all know from your background how that's been used in a number of conditions over the years. And there are 12 candidate intranasal products, which I've listed there, the reference in the Lancet, that are coming through. Now, you'll remember that the difficulty here is that you've got to look at mucosal immunity, which is not as easy as looking at what happens when you introduce the protein intravenously or intramuscularly. So the response may not be as good. And equally, people vary in their secretions. People get colds and flu and so on, and therefore you don't necessarily get the delivery of the vaccine adequately that way. So I would imagine that we'll see the results of at least 2 or 3 of those in the next year. And they're not all going to be successful, but that could be another either add-on or alternative to the current injectables. Next, please. Now, just to remind you from your student days, you know, viruses aren't like bacteria that can grow in soup and broth and so on. They can't reproduce by themselves, and they have to actually get inside a living cell, your cells or animal cells, and turn your cells into virus-making factories. So they have the instructions of how to copy themselves, but they don't have the tools to do that. Next, please. So here's the diagram of basically what's happening. Here's that little SARS-CoV-2 COVID-19 virus that gets into your body, and it's then got to find a way of living. So it's got to attach itself to a cell. And then once it does that with the ACE2 receptors, it goes inside the cell, opens itself up, and starts to reproduce. It starts to reproduce the RNA, DNAs, and then it wraps itself up and assembles itself into a new little virus, which matures, and then it's released to go and do its dirty work somewhere else. And thousands of those can be made in your cells. So the trick with a vaccine is to stop it adhering to the surface of your cells, like a policeman, if you will, to arrest it. Next, please. Here we are. There's the virus, you'll see on the right, that's like the spikes that are on the surface of the virus. And they want to get attached to the surface of the cell through this ACE2 receptor, which is activated by a protease. So if we can get the body's immune system to respond to that spike protein and grab hold of it by forming an antibody, then we can actually prevent the inclusion. Next, please. So that's what the vaccines do. They make antibodies as the policeman to arrest the spikes and so on. But ideally, what you need is to kill them, not just to arrest them, but to kill them. And the body, of course, makes T cells to kill aberrant cells, whether it's cancer cells or whatever. And frankly, right now, the vaccines we've got and the antibodies that we're using are not very effective against these producing these T cells. Next, please. So what's happening is whether you use the traditional weakened inactivated virus like the Sputnik or the Sino variety, or the newer RNA DNA vaccines, the responses are really looking at that spike antigen. And that's not going to give you the long-term cross-reactive memory T cell response that really in the end is going to be the way forward. Next, please. So a number of us are working on universal or pan-coronaviruses. My own company is TechImmune in California. And what we've done is to identify sequences in these coronaviruses that are common to millions of them. So they're conserved. So in fact, you know, if we make a vaccine against those, there's a very high chance that it will activate the CD4 plus and CD8 plus T cells, as well as arresting the virus that way. These different companies working, or universities working on them, all of them right now are in the preclinical phase, and we expect we would go into clinical studies in 2023, and of course safety studies. So we should see, hopefully, something completely different emerging, which is a pan-coronavirus in 2024, 2025. But meanwhile, we have to live with the vaccines we've got. Next, please. And there are so many in clinical trials. This is the very latest data from a couple of days ago from the New York Times review, which keeps it up to date. And you can see how many, there are 119 candidates in clinical trial and 75 more in preclinical studies. We're going to see failures, and you can see on this how many have been abandoned because they simply don't work. But no question, there will be emerging some of the current, should we say, spike-only kind of vaccines in the next 12 months to add to the Johnson & Johnson and the Pfizer, BioNTech, and the AstraZeneca, Oxford, and the Sino, and so on. But then I think the real breakthrough is going to be with those in a couple of years' time, which will be more universal. Next, please. There's quite a lot of stuff around on using just antivirals in nasal sprays. This particular study here, which you can look up in mice, shows it works pretty well. But I mentioned earlier all the problems about intranasal sprays. But it does seem that there could well be room for a nasal spray, which isn't a vaccine, but actually would be an antiviral approach to this condition. Then you can imagine how cheap that could be and how universal it could be used. Next, please. Here's one in a small company from the UK. And what they've done is to take interferon, which you know is the one that interferes with viral and cell replication. And they've made 11 changes in the amino acid sequencing in the receptor binding regions, so that rather than the body think, oh, I can cope with that, I don't need that, you've got 11 different changes. And so This is an intranasal product that is being developed. It's now in clinical phase 1 studies, and we'll know whether that's going to come through within the next 12 months. Next, please. Meanwhile, you'll know if you walk into pharmacies anywhere in the world, people are offering these kind of intranasal sprays, powder or liquid, and they're just antibacterial, quite frankly, antiviral, not very effective. And sometimes the claims that are being made on the web are really not acceptable. In fact, the regulatory agencies have had to write to the companies saying, please stop making those claims. So look, no question in my mind that keeping yourself distant from the virus with a mask and distances and using anti-microbial sprays is all good stuff because it prevents the spread. But these aren't going to cure, they're not going to treat the condition once it's there. But nevertheless, there is room for them, provided we don't make the customer, the patient, feel that they are cures. Next, please. I've come across this, which I'm working with this company Ondine on, this Canadian company, and they have over the last several years used this technology to prevent infection spread within hospitals in major cities in Canada. Basically, you take a methylene blue formulation, it's a very special formulation, and dab it with a swab into your nostrils, and then put these little electrodes in your nose, wearing goggles, and with a quick burst of a laser light, it activates the oxygen radicals, and then you kill all the materials in the nasal septum. Works extremely well, prevents, no question, prevents spread in wards. So if you're a professional pharmacist working in a ward, a doctor, a nurse, or whatever, just doing this for yourself could be very valuable in preventing you getting that. In fact, it's being used in a meat processing factory. All the workers there are using this to actually allow them to go to work without having the problem of being concerned about infection. But there are studies now, which I'm looking at the initial results of, to treat infected patients. And they're early days, but I would think within the next 6 months or so, we'll know whether this approach and how long you have to use it for will allow you to treat patients with laser light therapy and methylene blue. Next, please. We've not had much success with neutralizing monoclonal antibodies. I mean, if the body is not able itself to make the antibodies that you want it to, then you can get them, grow them in the lab and inject them that way. Next, please. And the problem is that just like with the vaccines, that these viruses with the spike proteins find their way around these very quickly. But there is some data now, at least one international consortium is actually mapping the binding of these antibodies that we've got, and therefore looking at how we might find different antibodies that could be more universal. But again, if they're only going to be effective against the spike, they're not necessarily going to be good for long-term use. In other words, we may have to make more and more antibodies each season. Like we make flu vaccines every season, we may have to make corona vaccines every season until these universal vaccines and antibodies come along. Next, please. Obviously, antivirals drugs are, we've heard from Dr. Hasan, are really important. And what we've been doing is to try to repurpose or use existing ones. But, you know, they're not designed specifically against the coronavirus. Next, please. So if you look at that diagram again of how that coronavirus hits the surface, adheres to the surface, gets inside the cell, starts to unravel itself, reproduces itself, and then forms more viruses. You can see where these little red boxes are, that there's different areas where we can use our knowledge of virology and antivirals, different antivirals, to actually develop small molecules and some larger molecules that could inhibit the transcription, could inhibit the proteome formation, inhibit the capsid going around it, or prevent it maturing, and then block that cycle. Next, please. Tried a lot of these. If you look at this bottom area here, that 54 of the ones we tried were just failed, just totally ineffective. But there are still many antivirals in studies, late stage, and I'm confident some of these will come through. The question that Dr. Hasan posed, of course, is very important. When do you use them? Only when the patient is definitely hospitalized, or when they're just testing positive at home? And how do you use it? And all those are big questions that each of these different new antivirals will have to go through. Next, please. Basically, the two that are really looking interesting to me are these ones that dependent upon RNA polymerase, in other words, stopping the messenger RNA transmitting the knowledge of how to build this new virus, or blocking that protease, like pexlovite does that. And so lots of those candidates now coming through, and I would expect that certainly the Shionogi one we'll see the data within the next 6 months on that. But I think gradually we'll see more of those coming through. They won't all work, but I think we can find them much more effective than the current ones which Dr. Hasan talked to. Next, please. So there are 982 products in clinical study, of which 430 antivirals. So there's hope that on a statistical basis, if nothing else, that some of those will come through. Next, please. Finally, I mentioned last time we had one of these seminars, and some of you were at that, what the G7 ministers did, and this was a mission that the UK government led, and now the German government have picked up. I was very fortunate to be part of that, is to say, let's work on the basis of looking at globally at what's coming over the horizon in terms of potential pandemics and coronavirus variants. So the WHO have set that up to monitor what's going on internationally. And meanwhile, we're creating 3 different groups, one to find diagnostics, one to find therapeutics, and one to find vaccines. And these are consortium of individual companies, universities, and government researchers. And the idea would be for therapeutics, for example, for antivirals, that we'll pick 25 different potential pandemic candidates, do all the work that we have to do in the animals and the cells, and then make our phase 1 studies in humans, and then put them on the shelves, oven ready, so the moment we get a new virus described that could be pandemic, within 100 days, we'll have it on the market. And in the meanwhile, that absolutely requires us to get our international clinical trials capability organized and harmonized regulation, and an equitable and sound way of distributing these to the developing world. So I hope that's given you an overview that we are working very hard in this area. Some results in the next few months, but most of them about a year or so away. Hope that helps. Thank you.
Thanks so much, Professor Jones, for that very informative view of what's in the pipeline, lots and lots in the pipeline. I'm particularly excited about the more universal vaccines. So let's hope we see them, as you say, within the next couple of years. So it's now time for our panel discussions. If I ask, can ask speakers to share their screens, and I'll just have a look. I think we've got a couple of questions in the question and answer box so far. Please do keep your questions coming in. But if I go to the first one, and I'll direct this to both Dr. Hasan and Professor Jones. Are these different therapeutic agents that you mentioned, can they be effective for all the different variants of COVID-19, or what is more efficacious?
Shahzad, would you like to pick that up for the existing ones you mentioned?
Yeah, for vaccines, we know that some vaccines, they are effective against certain variants, but in case of therapeutics, they have like a fixed design, formulation, we can't change much. It depends on, the trials, how they conduct the trial for which group of patients. So it's very difficult. That's why most of these antivirals are not effective, especially for the new variants, if you look at the evidence. So it's very difficult to come up with a therapeutic agent, antiviral, agent, which is effective against, the different variants. That's why we have some positive results and negative results in RCTs. If you look at even if you look at the last year trial, they are totally different from the new trial, the recent trials. So results are different, and we are having new antivirals coming in the market. So it's very difficult actually to predict or to say that, one antiviral is effective against all the different variants.
Yeah, so I think that's exactly right, that these were picked out of the historical bag of products that worked in flu or whatever. So you don't expect them really to be very effective against all strains. The ones that I've mentioned, there's hundreds of them in development, as I mentioned, they are being picked on the much more broad spectrum effect. The trouble is that these viruses are very clever, and these, say, the new South African 2 variant strains, you've designed it against all the current known variants, and then suddenly another one occurs. So the answer is going to be yes, the new generation will be much more effective against different variants, but there's still going to be some errors. And I think that will then come to combination therapy, because that's how it generally works with HIV and so on. The second question is about the side effects and adverse effects of anti-inflammatory drugs. Dr. Hasan, this is your field, isn't it? What do you think about the prednisolones and so on, the side effects?
Yeah, that's, for dexamethasone, the corticosteroids, I mentioned that, specific one is effective against the severe or moderate form of COVID-19. Yes, there is a risk of hyperglycemia and other things, associated with the use, but if they are using it for, low dose dexamethasone, so the risk of side effects is much lower compared to, the other drugs. If you compare tocilizumab, they use it as a single dose or 2 doses for the treatment. And even in RCTs, they haven't reported any severe, serious side effects or ADRs with the use of anti-inflammatory drugs. With the remdesivir, yes, there is a risk of acute kidney failure and other, serious ADRs reported for remdesivir. But for anti-inflammatory drugs, tocilizumab is safe because they are using it as a single dose treatment or 2 doses or 3 doses maximum. For dexamethasone, they are using it, low dose 6 milligram per day. So there is a risk of hyperglycemia is reported in some trials, but you can manage with with the insulin and other drugs. But there's no severe side effects reported.
And I guess like all therapies, it's a balance between risk and benefit, isn't it? That if you don't use these anti-inflammatories, then the sequelae could be very dangerous, yes, not just in terms of the fulminant infection and inflammation, but the long COVID effect. So suppressing these is necessary, but there's a risk.
Okay, there's also another question on ACE receptor-based therapeutics.
Yes, well, I mentioned the ACE2 receptor, the port of entry or the port of attachment of the virus, and there's about 15 candidate drugs that are looking at that. None of them have yet got to human studies. They're all preclinical, but I'm sure 1 or 2 will get there. But no data yet on any human trials.
Dr. Hasan, are you aware of any data on that area, ACE receptor-based therapeutics?
No, I'm not aware of the new investigational therapies, but yes, with the previous, ACE inhibitors, there were some trials where they look at the, use of ACE inhibitors for the prevention of, preventing people from acquiring COVID-19 infection. But the results were not statistically significant. There were some trials actually designed to see, but I'm not aware of the new investigational therapies or any new product in the market.
Okay, great. Thank you very much. We've got a booster question, and I'll again direct this to Dr. Hasan and Professor Jones. What is your opinion on the need for another booster, a total of 4 vaccines per person? And how many more do we need to give?
Well, I'm about to have on Monday my 4th dose, so I'm really interested in this. Of course, it depends on which vaccine you had originally as to how long it lasts, but not just upon the vaccine, but you as an individual and your immune system's competence for that particular challenge. And of course, the variants that are in that mix. So we've been studying, mixtures. If you say, start with the Pfizer vaccine and the next one you give is the AstraZeneca one, or vice versa. And of course, we've been seeing a few side effect profiles coming up that way. But there's no question that all the data I've seen, and I'm sure Dr. Hasan has more knowledge than I have in the area, these are not lasting forever. And so getting a boost is really important. Now, we do that with flu every year, and we have to guess what variants are coming up every year to make new batches. And that's what we're doing right now with the existing vaccines. The next batch is made will have more spike protein and so on. But I feel that certainly 4 boosters necessary, and probably one every year. Shahzad,
yes, I completely agree with you, because it will be something like flu vaccine where we need annual, boosters, because with the new variants coming in now, so the situation in South Africa and also in China, in Shanghai, the cases are increasing with new variants coming in. So maybe it will be like annual, vaccines, like flu vaccines. Every year we have to take, and with we have the design, the pharmaceutical design, where we can change the variants. We can come up with new vaccines formulation every year to against the new variants. So we have the technology now with us.
And I guess surveillance is important, of course, to know which variants are around. And we have a question around the Omicron variant. And this is quite specific to the Johnson & Johnson vaccine, but how effective is it against the South African Omicron variant?
Well, it's a question of whether this is, I presume, the new variants, because there's no doubt, when we looked at the data in South Africa, for South Africa with the J&J, I mean, the vaccine is 85% effective, which is, pretty good stuff, really. But that was on the variants in that study, which completed last year. So I don't have any up-to-date data about these 2 new variants. Do you have that, Dr. Hasan?
No, there's no published data, but there are some press release, but there's no published data they have shared or published in any journal.
So we'll have to keep a watchful eye on that one, I guess, as we would with any other variants.
Yeah.
Final question we've got here is, what do you think about the rebound COVID from using Paxlovid? Either Professor Jones or Dr. Hasan, any thoughts on that?
I don't actually understand the question. What's?
Is there a rebound effect? I'm presuming that means some kind of rebound illness. Not sure what it means.
I mean, obviously, the infection you get, which starts respiratory and then goes to the nasal to lungs, then sparks off all sorts of other things in different parts of the body. If you can shut that down and get rid of the virus, you've still got the chance that in the longer term, those inflammatory responses that have been triggered will produce other symptoms and you get long COVID. But this looks to be specifically a question saying you might get some of those effects by using Paxlovid. I don't know. Do you know that, Dr. Hasan?
Yeah, I know. I think there were some concerns about, because they're using it for mild cases for 5 days treatment only. So there were concerns, some of the researchers, they, not in the form of a research article, but in the form of expert opinions. I read, there was a risk of, rebound infection with the use of Paxlovid because they're using it for mild cases and only for 5 days. But there is no research data to support this.
This is a classical thing in pharmacy, isn't it? That you prescribe a course of therapy, take one tablet every day for 2 weeks. People feel great after a few days and put the medicine in their cupboard for the next time. No, take all of the course of therapy. It's killing 99% of the germs easily. It's the 1% you've got to get for.
So there's a strong message. Complete the course, everyone. Okay, I think we've got through the questions in the Q&A box. I've just got one question just to finish off. And I know at the start of the pandemic, the WHO were obviously very concerned about an infodemic, and how we manage all this information and how we manage misinformation and all the myths you get. And I think what Dr. Hasan and Professor Jones you've presented today is all the evidence. And I suppose my question is, how do we counter? There will always be a little bit of misinformation about how best to counter that. Any sites you think are very useful around the evidence base, or any other advice you would give about keeping an evidence-based approach to the fight against COVID-19? So I start with Dr. Hasan.
Yeah, if you ask about my strategy, I wrote many letters to the editors. When I read something and I find something controversial, something which is not right in terms of the data presentation, in terms of the analysis, I always write letter to the editor. And I wrote 50 plus letter to the editors in last 2 years, trying to convey my concerns, my understanding to the authors. They publish something which is not right, or I feel that is not correct. So even the journals are not like big journals. Sometimes the journals are like, very new journals, but they are index journals. So if they publish something, I write to them and I ask questions. And for the readers as well, because they publish something like vaccines. I remember in one of the vaccine journals, they published that Pfizer vaccine is not safe because there is a risk of death and mortality, increased mortality. So I wrote to the editor, and before they accept my letter to the editor, they actually retract the article, if you remember. So one approach is to write, a letter to the editors and counter the articles, the information they share. Or if it's not possible, that is on individual level. If more people are writing, then it's better. And another thing is that when we are reading articles or anything, we look at the quality, we look at the discussion, we look at the data. Is also important as healthcare professionals. We know how to synthesize the evidence, how to assess the quality of the evidence. And with social media platform, because of WhatsApp groups and Facebook and Twitter, people, they share information before before actually they read the complete article or understand the article. They share and also participating in groups that are, for example, the anti-vaccine groups. So sometimes I go and attend their their sessions and see what they are discussing. So I try to participate and go and discuss with them, okay, why you are criticizing this particular thing or product. So it's important you go and attend their sessions, and there are audience. So they will listen to your discussion and your points as well. So also by participating, in different sessions, go and share with people, there are large audience, in those sessions, so you can go and participate. So there are few approaches where we can contribute. As an individual, we go and we write letter to the editor. We can join sessions where they're talking about controversial topics and contribute. Yeah. So these are the few options that I approaches that I use.
That sounds very robust and very worthwhile, the work you're doing. Thank you, Dr. Hasan. Professor Jones, any advice?
Well, briefly, obviously, bad news sells newspapers and television programs, and the press journalists love to make sensational things. And it's so annoying when the people who receive that information don't have enough knowledge of science to understand the implications. So I think it behoves us as healthcare professionals to actually fill that gap. Many of you now have enough knowledge in this area to be able to put together small presentations and so on, and go into the communities and talk to people. I do this with some of the groups in England, some of the old folks groups or whatever it may be, just to try and have that dialogue directly with them and then answer their questions directly. But I think, to go back to the point I made about risk and benefit, inevitably taking any medicine is a risk, and we have to have a balanced conversation about what benefit it is and when we should do it. So thank you all very much for participating, and please go out there and spread the word.
Thanks, Trevor, and thank you both for the really expert advice you've been given. I'll just going to close off the event now. A huge thank you to our speakers for their really great contributions to today's current approaches to the fight against COVID-19, but also future approaches. I've certainly learned a lot. I hope you as an audience have been armed with the latest evidence and you're ready to go out there and give a balanced view wherever you are asked for your expertise on these medicines and vaccines. Our next event in the series is Managing Workforce Demand in a Pandemic Now and in the Future. That will be on the 14th of June and at the same time. Just a reminder, if you want to revisit this event and any of the events on the FIP calendar, this is where you can go to visit that. There's plenty of other events going on as well, which we hope will find interesting. So that just leaves me to say thank you so much for attending today. Always a pleasure having you, and enjoy the rest of the day wherever you are in the world. Goodbye.
Bye now.